Hi everyone, today I'll be walking you through the process of cell respiration. This is a follow along with me drawing activity, so be sure you have an 8.5 by 11 sheet of paper on hand. This is the horizontal orientation. Make sure to allocate about a third of the bottom section of your page to section into 6 boxes, 6 rectangles, so that you can write the products or the outputs of every process as we move along from glycolysis all the way to oxidative phosphorylation. Speaking of that, I'm just going to label the steps here. So first we have glycolysis, and then we'll look at pyruvate oxidation, and then we have the Krebs cycle, also known as the citric acid cycle. So a way that I remember the citric acid cycle and the Krebs cycle as being part of cell respiration instead of photosynthesis, since photosynthesis has this Calvin cycle that's, I don't know, just maybe because they all start with like a K sound, or maybe they have like a C in their name. I just get these three all mixed up. So I remember that Krebs has an R in the name, and citric acid cycle also has an R in the name. So I associate these two with cell respiration. Whereas the Calvin cycle doesn't have an R in the name, I'm just going to remember it's with photosynthesis. And then I am going to, there's this process called oxidative phosphorylation that is the coupling of two processes that I'm going to break into 2 steps called the electron transport chain and chemiosmosis. So we're going to look at these two separately, but they are both part of oxidative phosphorylation. So down here, I'm going to make an indicator. That these two are known as oxidative phosphorylation. Normally, you would see cell respiration broken up into number 1, the first step is glycolysis, number 2 is the Krebs cycle, number 3 is oxidative phosphorylation. But I have broken these down into smaller components, looking at some of the intermediate processes so that we can fully understand how a glucose molecule's energy is converted to ATP. And this last box over here is going to be where we keep track of the ATPs produced in each of these processes. So outside of the mitochondria, we have the cytoplasm. And as for the parts of a mitochondria, I'm going to use a different color here. Let's use brown. I'm going to have, this is the outer membrane. Remember that a mitochondria is a double membrane organelle, and it also has this inner membrane that I'm just going to outline. The region between the inner and the outer membrane is called the intermembrane space. So I'm going to write that down here just because I have more room compared to the top. This is the intermembrane space. And then this big region in the center over here, that is the matrix of the mitochondria. I'm going to label that on top over here. This is quite important. Now, going back into the cytoplasm, this is where glycolysis is going to take place. Let's start with a glucose molecule. We all know that cell respiration starts with glucose. It is a 6-carbon molecule, and for glycolysis to take place, we need to invest 2 ATP molecules into these reactions. And we're going to label this investment. So this is a 2 ATP investment. Products of this glucose molecule, we're going to write down here. Glucose is going to be split in half, creating molecules called pyruvates. And these are 3-carbon molecules, and you'll have 2 of them, as you can see here. you're going to have these electron accepting molecules called NAD+, coming in to extract and take electrons from glucose. When it takes an electron, it's going to gain an electron, and we call that becoming reduced. So NAD+ is going to become reduced and become NADH when it gains 2 electrons as well as a proton. And you will have 2 NADH molecules. You are also going to create 2 water molecules. This occurs in the 9th step of glycolysis. And you will create 4 ATP molecules. This is by substrate level phosphorylation. And now we're going to do an accounting of all the products that we've created in glycolysis. So starting with 1 glucose molecule that contains 6 carbons, now we have 2 pyruvate molecules, each containing 3 carbons. So far this makes sense. It's like the glucose molecule was cut in half. We also have these electron carriers, these reduced electron carriers called NADH. We have 2 of these. They are each carrying 2 electrons and 1 proton. We also have water created. We have 2 water. And we also have ATP. So technically we created 4, but we have to remember that 2 molecules of ATP were invested. So we have to take that into consideration when we account for the net ATP generated by glycolysis. So the net ATP is 2. Since we created 4, but you know what? 2 was invested to get this process started. Now we have these 2 pyruvate molecules that we're going to follow into the mitochondria. And I'm going to use a different color for that. I'm going to follow just one of these pyruvate molecules and redraw it over here. This is the 3-carbon pyruvate molecule. An enzyme called pyruvate dehydrogenase is going to carry out pyruvate oxidation. So pyruvate oxidation is, again, the extraction of electrons from pyruvate, so making pyruvate lose more of those electrons. And it's a 3-step process that pyruvate dehydrogenase assists in. First, it's going to take away a carbon from pyruvate, which leaves 2 carbons, which we're going to call an acetyl group. There is the addition of a coenzyme A, which is what makes acetyl CoA. This molecule now can then enter the citric acid cycle, also known as the Krebs cycle. We're going to be looking at that later. There is also another product formed from the reduction of another NAD+, so that it gains 2 electrons. It extracts 2 electrons from this pyruvate molecule, as well as 1 proton from this matrix to create NADH. So altogether, the products of pyruvate oxidation are, you get acetyl-CoA that is going to go into the Krebs cycle, and you have 2 of these because you start out with 2 pyruvate molecules. Each one of these pyruvate molecules is going to undergo pyruvate oxidation via an enzyme called pyruvate dehydrogenase. So now you have 2 acetyl-CoA. You're also going to have 2 carbon dioxide, as well as 2 NADH. I want to emphasize once again that this is showing the path of 1 pyruvate molecule. Remember, 2 pyruvate molecules are created from glycolysis, so it's actually this reaction times 2, hence why all of these products are multiplied by 2. Down here, I'm just going to put a note that the enzyme that catalyzes these 3 steps is called pyruvate dehydrogenase. Pyruvate oxidation is the first step in which carbon dioxide is released in cell respiration. Carbon dioxide is also released in the Krebs cycle. And so far we have NADH storing energy extracted from the pyruvates. This acetyl-CoA contains very high potential energy that it can bring into the citric acid cycle. So now the citric acid cycle is going to be in pink. And I'm going to draw a sort of cycle with an arrow going in the cyclical direction. And there is a 4-carbon molecule called oxaloacetate. Oxaloacetate is regenerated in this citric acid cycle. It's going to combine with these 2 carbons from acetyl-CoA. The coenzyme A is going to be released, and the combination of oxaloacetate and the acetyl group is going to create a 6-carbon molecule. That 6-carbon molecule contains a lot of energy, lots of electrons that can be extracted by NAD+ and FAD, which will become reduced into NADH and FADH2. We have 3 NAD+ molecules that are going to each accept 2 electrons plus 1 proton from the matrix to create 3 NADH molecules. In the process, they will also release carbon dioxide, and they'll release 2 carbon dioxide for every cycle. Also, there is GDP, which can be phosphorylated through substrate level phosphorylation to make GTP. This is what occurs most commonly in animal tissue cells, and this GTP can then be converted into ATP. In certain plants, bacteria, and other animal tissue cells, there is also ADP that can directly be converted to ATP. So this is only one molecule of GDP becoming ATP, or ADP becoming ATP. Next, we have another oxidized electron carrier called FAD. It's going to become reduced once it gains electrons and become FADH2. The difference between FAD and NAD+ is that FAD is going to gain 2 electrons and 2 protons, whereas NAD+ is going to gain 2 electrons and only 1 proton. Hence, it becomes neutralized as NADH, and FAD gains these 2 hydrogens. Something else that I want to add here is that in the citric acid cycle, there is an input of water at this point. So a water molecule is input. That's going to come into play later. The reason why I'm including this water molecule right here, I'll explain later. But now we're going to look at the products of this Krebs cycle, or this citric acid cycle. Go down here and now calculate how many total products were created. Starting with NADH. This is the reduced form, meaning it has gained electrons. It's currently carrying 2 electrons and 1 proton. So 3 NADH molecules were created with every acetyl-CoA. You have 2 acetyl-CoA molecules, so you're going to have to multiply the number of the products by 2, which is going to give you 6 NADH molecules. Now we're going to look at GDP, ATP. We have GTP that can become ATP, and only one of these is generated with every cycle. You have 2 acetyl-CoAs, so 2 cycles will get you 2 ATPs. And again, this is by substrate level phosphorylation, same as the one that occurred in glycolysis. Next, we have another reduced electron carrier called FADH2. Only 1 molecule is generated in the citric acid cycle right here. And if we have 2 acetyl-CoAs, that means we have 2 FADH2 molecules. I also want to point out that I forgot about the carbons over here. For every cycle, 2 carbon dioxide molecules are generated. So we have carbon dioxide times 4, because 2 acetyl-CoAs, 2 cycles, 2 times 2 carbon dioxide formed. And we also have something called oxaloacetate that is regenerated every citric acid cycle. So every cycle, these 4 carbon molecules are regenerated, ready to accept this acetyl group from acetyl-CoA. So next we have this electron transport chain. But before we go into that, I want to do some accounting and just take note of all the very high energy electron carriers that we have so far. So using a different color, I'm going to use a bright yellow color. I'm just going to go through and circle the reduced electron carriers in the mitochondria. So you have, I'm not going to circle the number, I'm just going to circle the molecule name. So you have NADH, you have FADH2 from the Krebs cycle. From pyruvate oxidation, you have this NADH. What about these 2 NADH molecules sitting in the cytoplasm from glycolysis? NADH is a molecule that cannot enter or pass through this mitochondrial membrane. So what these NADH molecules have to do is that they have to donate their pairs of electrons that I'm drawing right here. They have to donate their pairs of electrons through the membrane, and they have to donate their pairs of electrons through the membrane, and those electrons will be accepted by other electron acceptors. So another NAD plus or an FAD molecule. If both of these pairs of electrons are accepted by NAD plus, that gives you NADH, and you would have 2 molecules of NADH. If both of these pairs of electrons are donated to FAD, that would make FADH2, and you would have 2 molecules of FADH2. So depending on which electron acceptor is receiving the electrons donated by the NADH molecules created from glycolysis, there can be varying numbers of ATP produced. These electron acceptors carry different amounts of energy that can be translated to the amount of protons that can be pumped from the matrix to the intermembrane space, hence the amount of protons moving through ATP synthase to generate ATP. And we're going to do some accounting on that later. Here I just wanted to point out, this is how the electrons generated by glycolysis are going to enter the mitochondria. I'm going to change my color now to this one that I was using to keep track of all the electron carrying molecules. For the rest of this process, I'm just going to track the movement of this NADH molecule. So now I'm going to add numbers to calculate exactly how many of each electron carrying molecule I have in the mitochondria. Let's pretend that we get NADH, and we're just going to follow this one through the rest of the process of cell respiration. We have these 2 NADH molecules carrying their 2 electrons on each molecule. We also have this NADH molecule that carries 2 electrons. We have this FADH molecule carrying 2 electrons. This NADH molecule also carrying 2 electrons. So these are the sources of very high energy electrons that can be donated to the electron transport chain. How many NADH molecules do we actually have? So if we look down here in our first 3 boxes, let's box the number of NADH molecules that we have. We have 2 NADH molecules from glycolysis that is represented right here. And we have 2 NADH molecules from pyruvate oxidation. So I'm just going to write times 2 there. And we also have NADH from the Krebs cycle, and this time we have 6. So I'm going to write times 6. So the total number of NADH molecules, the total number of these reduced electron carriers is 10. We have 10 NADH molecules, each containing and carrying 2 electrons. What about FADH2? Right there. So you can see that FADH2 is only generated in the Krebs cycle, and 2 is generated per glucose molecule. So we'll have, okay, so starting with the NADH is going to drop off or donate its electrons to the first protein of the protein. This reduced electron carrying molecule is going to donate its electrons to the first protein complex of the electron transport chain. So I'm going to draw these electron transport chain complexes. Here's the first one. Here is the second one. It's also going to contain a molecule, a non-protein molecule called ubiquinone that I'm going to abbreviate with Q. And here is the third protein and the fourth protein. And while I'm at it, I'm also going to draw ATP synthase. Over here. So this is protein complex 2, protein complex 3, protein complex 4. All right, now I'm going to change my color so that I can highlight the path of these electrons. I'm going to use this color. Okay, so I'm going to just highlight these electrons again. NADH is carrying 2 electrons. It's going to drop off both electrons in this first protein complex. These electrons are going to move from protein complex 1 to protein complex 2. Ubiquinone is going to carry it to protein complex number 3. It's going to get shuttled to protein complex number 4. And then there is a very electronegative molecule at the very end of this electron transport chain. This entire complex is the electron transport chain. It does not include ATP synthase over here. So at the very end of this electron transport chain is oxygen. Oxygen is very electronegative, and I'm just going to use this space in the middle over here to show you how the electrons are going to help make water with oxygen. So an oxygen atom has 6 electrons, 1, 2, 3, 4, 5, 6. This is a Lewis dot structure. When these 2 electrons are shuttled through this electron transport chain, the electrons are going to help make an octet with one of the oxygen atoms. So now you have these 2 electrons making an octet. Remember, in the matrix, there is also protons floating around. And these protons are going to combine with this oxygen molecule. So that proton moves over. Here we have a water molecule. That's not the most accurate shape of a water molecule. It's technically bent. But you can see H2O, it has 1 oxygen, 2 hydrogen atoms. And it has this perfect octet. So this is how water is created from these electrons moving down the electron transport chain, combining with half of an oxygen molecule. Oxygen molecules exist in diatomic forms, meaning that in nature they exist as 2 oxygen atoms bonded together. So we only need one of those oxygen atoms, hence I added this 1/2 sign in front of this oxygen molecule that contains 2 oxygen atoms. So now we know the path of these NADH electrons. What about FADH? FADH is going to donate its electrons to protein complex 2. Reason being that FADH is less able to donate electrons. It's going to donate electrons at a later stage where there's less free energy. So the electrons donated by FADH2 are going to enter over here. And again, they're going to follow this electron transport chain towards water, where they're going to make another water molecule. So it takes 2 electrons, 1 oxygen atom, and 2 protons to make a water molecule. Knowing that information and thinking how many total electrons are fed into this electron transport chain when we have 10 NADH and 2 FADH2, we're able to calculate how many water molecules are generated from this electron transport chain. So if you have 10 of these molecules and they each contain 2 electrons, just doing the math in the upper right hand corner, you have 10 times 2 giving you 20 electrons from NADH, and you also have these 2 FADH2 molecules each carrying 2 electrons. That gives you 4 electrons. So you have a total of 24 electrons moving through this electron transport chain towards oxygen. So if it takes 2 electrons to make a water molecule, that means 24 electrons divided by 2, you're able to generate 12 water molecules from 10 NADH and 2 FADH2 molecules that you commonly see being used to depict cell respiration. That is 1 glucose molecule, C6H12O6, plus 6 oxygen molecules is going to make 6 carbon dioxide plus 6 water molecules. If you take a look at this equation, this is a balanced chemical equation. And let's take a step back here. We know we have a molecule of glucose, and we can see here there are 6 carbon dioxide molecules produced. Look at our products down here. You can see that 2 carbon dioxide molecules was produced via pyruvate oxidation. 4 molecules of carbon dioxide was produced via the citric acid cycle. Okay, so this makes sense in that we have 6 carbon dioxide molecules. So now let's look at oxygen. If we have 6 molecules of oxygen, so 6 times 2, that is, you have 12 oxygen atoms. So far it makes sense. If you have 12 oxygen atoms, I'm just going to write that right here. If you have 12 oxygen atoms and you have 24 of these electrons, you are able to make 12 water molecules. So why is this only showing 6? Why is it that a lot of equations showing cell respiration 6 water molecules as the products? So to answer that, I'm going to write a different equation of cell respiration. You start out with, again, 1 glucose molecule. You also have 6 oxygen molecules. This time you are adding 6 water molecules. Your carbon dioxide molecules don't change. And now you can see how your 12 water molecules fits in here. So this equation is actually the true equation of cell respiration. This equation that you see up here, this equation up here is the net equation, which means that the water molecules invested into cell respiration has been accounted for and subtracted from the total number of water molecules formed, which gives you 6 water molecules. And that's what gives us this production of 6 water molecules. Now you might be wondering, what about glycolysis? Because in glycolysis over here, it generated 2 water molecules, and you can see that right here. Doesn't that go into the equation somewhere? Yes, it does. But remember over here in the Krebs cycle, I pointed out that water is also invested into this. But here in the Krebs cycle, I also told you guys earlier that water, but in the Krebs cycle, there is a point in time where, but remember that over here in the Krebs cycle, water is input into this citric acid But don't forget that we also input water into the Krebs cycle over here. There are other moments along the Krebs cycle where water is input, water is taken out, so they all kind of balance out. And in this case, the two water molecules generated in glycolysis are balanced out by what would be two water molecules entering the citric acid cycle. So there would also be two water molecules for the two acetyl-CoA molecules entering the citric acid cycle. So in this way, the water molecules featured here are going to cancel out. So the remaining water molecules that are taken away here to give us 6 water molecules as a net product, it's not illustrated in the textbook. It's information that you don't have to know in detail for the scope of the AP Bio course. But just so you know, this is why when you mathematically calculate all the electrons, all the oxygens, you'll realize that you end up with 12 water molecules. When you see equations with 6 water molecules, now you know what's up. 6 water molecules were used up in the process of cell respiration. Okay, so now that we've cleared up what can be a common question that arises when students try to calculate exactly all the products that are generated from cell respiration, we got water down. So we're going to come down here to the output section and fill in the outputs for the electron transport chain. So with the input of these reduced electron carriers NADH and FADH2, something I forgot to draw earlier is that when they deposit or donate their electrons, they become oxidized. And their oxidized form is NAD plus and FAD. And I'll also write the number of those molecules. So you would have 2 NAD plus, 2 FAD. So this is the oxidized form. Just going to write that as a little note. You also have FAD that is also oxidized. You have 2 FAD molecules. Also, you have created, remember, water. You have 12 water molecules, but I want you to write that the net water molecules generated is 6H2O. Okay, so this is the electron transport chain. Next, we are going to look at the second half of oxidative phosphorylation, and that is chemiosmosis. Chemiosmosis is basically the movement of chemicals. In this case, we're going to be looking at protons. Osmosis, you should think of passive transport, diffusion, the movement of water. In this case, it's the movement of these chemicals, these protons, down a concentration gradient. What concentration gradient, you might be wondering. We're going to be making that in a moment. So these protons, some of which are generated from the oxidation of NADH and FADH2. So the oxidation of NADH is going to generate 1 proton, whereas the oxidation of FADH2 is going to generate 2 protons. And the protons in the matrix, how they're going to move from this matrix area into this intermembrane space is that as these electrons are being pulled through these proteins, they're releasing energy in these proteins, and that energy released from these electrons are going to help pump the protons across the membrane. So that's going to happen in proteins 1, 3, and 4. Protein 2 is not going to pump any protons across the membrane. It's mainly serving as an electron acceptor for FADH2 to drop off its electrons. And protein 4 is going to also pump protons across the membrane. So there's going to be an accumulation of these protons, which over time is going to mean that the proton concentration in the intermembrane space is higher than the proton concentration in the matrix. So here we have high concentration. In the matrix, we have a low concentration of protons. Embedded in this inner membrane of the mitochondria is this ATP synthase. This ATP synthase is going to act as a channel through which protons can diffuse through. So these protons are going to want to move from an area of high concentration to an area of lower concentration. This proton gradient that has been generated is going to allow these protons to diffuse through ATP synthase by the channel provided. And in the process of that diffusion, it's going to rotate this rotor that is part of ATP synthase. As this rotor is rotating, zoom in here, it's going to help phosphorylate ADP. So we have an ADP molecule that is going to be phosphorylated to become ATP. So this entire process right here, where protons are diffusing through this ATP synthase, turning this knob down here so that ADP can be phosphorylated, meaning a phosphate group is added to ADP to create adenine triphosphate. This is the process that's going to generate the most ATP in cell respiration. So now if we just zoom out, this is the entire process of cell respiration. This area down here is the most important. Chemiosmosis is going to generate the most ATP. Altogether, the electron transport chain over here, coupled with chemiosmosis, is known as oxidative phosphorylation. Oxidative meaning it involves the losing of electrons. So the electron-carrying molecules, when they become oxidized, they have lost or donated their electrons to this electron transport chain. The movement of these electrons down this chain is going to pump protons from one side of the membrane to the other. So that pumping from the matrix to the intermembrane space, resulting in an accumulation of these protons in the intermembrane space. Movement of protons from high to low concentration is the basis of chemiosmosis. And through this chemiosmosis process, it's going through ATP synthase. ATP synthase has this rod connected to a knob down here that is going to phosphorylate ADP into ATP. So this entire process of ATP production is known as oxidative phosphorylation. And this is different from the other ways in which ATP was produced in the citric acid cycle and in glycolysis. Because in the citric acid cycle and glycolysis, they use substrate level phosphorylation, which does not generate as much ATP as oxidative phosphorylation. Okay, so thinking about the words oxidative and phosphorylation and how they tie together the electron transport chain and chemiosmosis, oxidative means involving the loss of electrons. So we know that these electron carriers, NADH and FADH2, lost their electrons to the electron transport chain, and the movement of the electrons down that chain is what produces this proton gradient as protons are pumped from the matrix into the intermembrane space. This proton gradient is what is going to allow for chemiosmosis, the diffusion of these protons back into the matrix, which is what's going to help phosphorylate, it's phosphorylation, the addition of a phosphate group to an ADP molecule. and that makes ATP. So the entire process of chemiosmosis can generate up to a total of 28 ATP molecules. Now you might be wondering, how exactly did I get this number? There are several scenarios. Remember, glycolysis had produced these 2 NADH molecules that could not enter the mitochondria. As a result, they had to donate their electrons to either an NAD plus or an FAD molecule. Depending on which of these electron acceptors becomes reduced, meaning they gain these electrons from the NADH molecules made from glycolysis. Let's put a line over here showing you. Okay, let's put a line over here showing you that this is the mitochondria, whereas this is the cytoplasm. Okay, so in the mitochondria, there is NAD plus, FAD, and they can accept these electrons from each of these molecules. They don't always move together. The pair of electrons can be donated to either an NAD plus or an FAD, regardless of where the other electrons are donated to, whether it's another NAD plus or another FAD molecule. So there's a lot of mixing and matching that can occur. There are 3 scenarios of combinations that can occur. So scenario 1 is where both of these NADH pairs of electrons are donated to NAD plus molecules, which means that you have created 2 NAD plus molecules, each carrying 2 electrons. This is the example that we looked at. So this is the example that we've been tracking throughout the electron transport chain, making water and pumping protons. So we'll start with this one. And this is 10 NADH. You guys should remember we got 10 NADH and 2 FADH2 molecules. Hence, we donated a total of 24 electrons into the electron transport chain. Now, looking at the protons, there's a specific number of protons that can be pumped across the membrane with every donation of an NADH or an FADH2 molecule. So here, just for your reference, as we do the calculations, one reduced molecule of NADH, when it donates its electrons, it's capable of pumping. When it donates its electrons, 10 hydrogen ions or 10 protons can be pumped across the membrane. Whereas for every FADH2 molecule, this reduced molecule can result in 6 protons being pumped across the membrane. Okay, and it takes 4 protons to make 1 ATP molecule via ATP synthase. Okay, so knowing this, we're going to do some calculations. We got 10 NADH, meaning that we can make 100 protons. 2 FADH is going to give us 12 protons, giving us a total of 112 protons. If it takes 4 protons to make 1 ATP molecule, I'm going to divide 112 by 4. That's going to get you 28 ATP. So this is where the number 28 comes from, shown in the diagram that we were just drawing. And this is scenario 1. So another scenario here would be if the pairs of electrons from these 2 NADH molecules, one was donated to another NAD plus, and another one was donated to an FAD, making FADH2. So in this scenario, you would have 9 NADH molecules and 3 FADH2 molecules. Okay, so it's like you gained 1 NADH from the electrons from glycolysis, the way they donated to this electron carrier, and this is the reduced version. And you had another pair of electrons that was donated to FAD. When you do the calculations for NADH, you got 9 NADH molecules times 10 protons for every NADH. You'll have 90 protons. For FADH2, you got 3 molecules. You are going to have 18 protons. You add this up, you'll get 108 protons. For every 4 protons, you make 1 ATP, which gives you a total of 27 ATP molecules. We have a third scenario here, and that is when, that's scenario 2. This one is scenario 3. My handwriting is just getting messier and messier. So in scenario 3, both pairs of these electrons from NADH are going to be passed on to FAD. So we're going to have 2 molecules of FADH2. So here we have 4 FADH2. Looking at this NADH, remember we got 2 NADH from Pyruvate Oxidation, and we get 6 NADH from the Krebs cycle. So that's what gives us our 8 NADH here. None of the NADH molecules produced by glycolysis has passed on their electrons to another NAD plus molecule. Instead, they have passed on their electrons to FADH2. That's what gives us 4 FADH2 molecules. So doing the calculations for 8 NADH, you're going to have 80 protons. For 4 FADH2, you are going to have 24 protons. Add that up, you are going to get 104 protons. Let's see how many ATPs are made from 104 protons. You are going to get 26 ATP. So these are the 3 different scenarios where variations from these electron carriers donating their electrons to either NAD plus or FAD. These are the different numbers of ATP that can be produced. NADH, I'm kind of covered it here. NADH, remember, it can deposit its electrons at a higher energy level at the first protein of the electron transport chain, so it's able to release more energy, hence pump more protons across the membrane. Remember, FADH2 enters and drops off its electrons at the second protein of the electron transport chain. Its electrons don't release as much energy as the electrons from NADH. Hence, it can only pump in 6 protons. So that's where the difference comes in. And looking at these numbers, the number of ATP molecules that can be produced from just oxidative phosphorylation where we're only using these high energy carrying molecules that deposit their electrons to move protons and then get our ATP from the protons diffusing back across the membrane. Oxidative phosphorylation is going to generate approximately 26 to 28 ATP molecules. 26 to 28 ATP. So in the example that I drew over here in this diagram, we only focused on using NADH, assuming that NADH is the reduced electron carrier accepting the electrons, both pairs of electrons, from the NADH made by glycolysis. In this last box, we're going to add up all the ATPs that we have created. So now we're going to go through our outputs down here looking for ATPs. We made a net of 2 ATPs via glycolysis. This was from substrate level phosphorylation. We also made 2 ATPs here in the Krebs cycle, also by substrate level phosphorylation. So I'm going to write here glycolysis made 2, the Krebs cycle made 2, and then oxidative phosphorylation makes 26 to 28. When you add that up, the least amount of ATP that can be generated would be 26 plus 2 plus 2, 30, 28 plus 2 plus 2 to 32. Okay, so this is the range of ATPs that can be created from cell respiration, 30 to 32. All right, so here's what it looks like overall. All right, so this is everything about aerobic cell respiration that happens inside the mitochondria. So you can see there's a lot of action in the matrix of the mitochondria. The inner membrane is also going to see a lot of action. Remember, the inner membrane of the mitochondria is very convoluted. It's very folded. It should be like this. This means that there's a lot of surface area all around the inner mitochondrial membrane for all of these proteins to reside, for the electron transport chain, for chemiosmosis, basically for oxidative phosphorylation to occur. You should also know that oxygen plays a huge role in cell respiration. The reason why we breathe in oxygen is so we have this electron acceptor at the end of our electron transport chain so that we can generate this proton gradient as electrons are pumping these protons from the matrix into the intermembrane space. Without oxygen, this electron transport chain would stop working. It's not going to be able to pull these electrons down the chain, and as a result, protons are not going to move across the membrane from the matrix into the intermembrane space. If protons don't move into the intermembrane space, we won't have this proton gradient that is formed where there's a high concentration on one side and a low concentration on the other. If we don't have that proton gradient, that proton concentration difference, ATP synthase would not run. ATP synthase would not be able to phosphorylate ADP. So ultimately, without oxygen, cell respiration would not work, ATP would not be created. If you draw this over and over and over and over again, I guarantee you will be able to draw this all from memory one day. Thank you for watching, and I hope that this video was helpful for you. And if you enjoyed it, please give it a thumbs up. Good luck studying for cell respiration.